What are SCN1A-related disorders? SCN1A-related disorders comprise a spectrum of different diseases. Most commonly, individuals present with the severe childhood onset epilepsy Dravet syndrome, while a smaller group of patients present with the less severe epilepsy called genetic epilepsy with febrile seizures plus, in short GEFS plus, or other diseases. In order to understand what SCN1A-related disorders are, we need to start at the very beginning, with genetics. DNA is a set of instructions contained within each cell of our body. Genes are the building blocks of life. They are just like chapters of construction manuals, but instead of constructing a car or a boat, genes contain the instructions for producing proteins that have different shapes and essential functions in the body. All cells in our body contain the complete manual. However, different cells build or express different chapters at different rates. This is known as gene expression. The SCN1A gene is expressed and functions predominantly in brain cells. It contains the instructions for a membrane protein, which means it is located at the border of cells, in this case neurons, which are cells in the brain. The protein produced by SCN1A belongs to the voltage-gated sodium channel protein family and allows sodium ions to travel through the cell membrane to pass on information. Expressed mainly at inhibitory neurons, these channels are crucial for initiation and propagation of action potentials along the neurons. Specifically, scientists have determined that SCN1A encoded channels are essential to the development of the brain and for the communication between neurons. In a manual, when instructions are missing, misspelled, or exchanged, the end product may end up different from what was initially intended. The same is true for genes. Even a small change in the instructions could cause the protein product to be partly or entirely changed or lost. This can have harmful effects throughout the entire body. These errors within genes are called genetic variants, and they occur all the time. When the SCN1A gene is mutated, meaning it has a disease-causing variant, an improper protein product might be created. Such an altered protein cannot form a proper voltage-gated sodium channel, which may alter the channel's ability to allow sodium ions to pass through. This then causes a disturbance in communication between brain cells, leading to disease. So, how can you tell if someone has an SCN1A-related disorder? Since different types of variants can lead to an SCN1A-related disorder, there are multiple presentations of the disorder, all of which are accompanied by various symptoms. In fact, even individuals with the exact same variant can exhibit slightly different symptoms. Pathogenic, meaning disease-causing variants in the SCN1A gene, have been most frequently identified in individuals with Dravet syndrome and GEFS+. Dravet syndrome, which is named after the French epileptologist Dr. Charles Dravet, who first described individuals with this disorder, presents in the first year of life with recurrent seizures, with or without fever, that are often prolonged and difficult to treat. Subsequently, other seizure types, such as myoclonic and atypical absence seizures, emerge, as well as long-term problems with learning, motor development, and behavior. In comparison, individuals with GEFS+ present with mild epilepsy and normal cognition. Apart from these more common presentations associated with SCN1A gene variants, there are also other, rarer forms of very severe early onset epilepsies described that present in the first few months of life. Researchers have compiled a list of symptoms seen in patients in varying severity. Epileptic seizures can be severe and life-threatening in individuals with Dravet syndrome, but can be much less severe and less frequent in individuals with GEFS+ phenotypes. This range of symptoms is called a disease spectrum. Furthermore, not all individuals with a pathogenic SCN1A gene variant have epilepsy. A small group of patients with SCN1A gene variants has been described that do not experience seizures, but a specific type of migraine associated with weakness on one side of the body called hemiplegic migraine. A spectrum of diseases could be associated with a single gene, such as SCN1A, because different variants can lead to different changes in the altered protein. For example, studies have shown that Dravet syndrome and GEFS Plus are typically caused by variants that lead to loss of channel function, while the early onset severe epilepsy is caused by variants that lead to gain of channel function. If an SCN1A-related disorder is suspected, it is important to perform comprehensive genetic testing to confirm the diagnosis. However, not all individuals with a typical Dravet syndrome or a GEFS Plus presentation might test positive for an SCN1A variant. This is likely due to certain types of variants that are difficult to detect using current genetic testing techniques. Existing treatment options for SCN1A-related disorders depend on both the clinical presentation of the individual as well as the functional outcome of the variant. Currently, SCN1A-related disorders cannot be cured. However, previous research studies point to a number of different medications that have been shown to be helpful for individuals with Dravet syndrome and GEFS Plus in improving some aspects of the disorder, for example, the seizures. Because Dravet syndrome and GEFS Plus are caused by different degrees of loss of channel function variants, sodium channel blocker medications should be avoided in these disorders, as they may worsen symptoms. Apart from treatment of seizures, therapy should also focus on managing the specific symptoms of the individual. For example, difficulties with motor development and behavior. Clinicians, patients, and their families should discuss the possible treatment options and tailor the treatment according to each patient's needs. For more information, please visit our SCN Portal, an interactive website for families, clinicians, and researchers dedicated to comprehending SCN1A-related and other sodium channel disorders. We encourage you to participate in SCN1A research to help investigate clinical and genetic findings of SCN1A-related disorders and collect data of affected individuals, paving the way to a cure. Please hit the like button if you have learned something new, and make sure to subscribe to this channel so you won't miss future uploads. Thanks for your support.